Hello everyone. So after a slight and short break, let's get back to our theoretical classes. Let's get back to our metabolism. But now we're shifting our focus from carbs, from lipids to proteins. So today and during upcoming several videos, we're gonna discuss about the protein and amino acid metabolism. So we are gonna start with the following the very well-known pattern. We're going to start with the first stage of catabolism. We're going to start with digestion of protein and absorption of amino acids. So let's make a short overview on the topic. So like we said for complex carbs, like we said for complex lipids, dietary proteins as well are so large and huge molecules, and they cannot be either consumed or absorbed in their native form. Rather, they have to be subjected to enzyme hydrolysis in gastrointestinal tract, during which large protein molecules are going to be degraded to the mixture of amino acids. The mixture of amino acids is then subjected to the process of absorption, by which amino acids are transported first to the enterocytes and then from enterocytes to portal circulation and then finally to tissue cells. In tissue cells, amino acids are organized in a so-called amino acid pool, from where amino acids can be used for either synthesis of new proteins or synthesis of non-proteinogenic molecules like glucose, acetyl coenzyme A, ketone bodies, and so on. Numerous proteolytic enzymes are involved in the process of protein digestion. They are synthesized in different tissues like stomach, pancreas, and small intestine. Digestion process can be divided in several phases. It can be described in a phase of listing events occurring in mouth, stomach, duodenum, small intestine, and so on. But it can also be followed in the following way. By number one, combined intraluminal digestion, which actually describes all events taking place in the lumen of the gastrointestinal tract. The second phase is so-called surface membrane digestion, which explains the events and chemical changes or physical changes which proteins, peptides, amino acids undergoing at the surface of intestinal epithelial cells. And finally, the last stage is called intracellular digestion in enterocytes, which actually describes how amino acids are absorbed to enterocytes and then secreted from enterocytes to portal bloodstream. But in order to get the story more understandable and more simple, I suggest that we start like we did for carbs and lipids. Let's start with the events occurring in mouth and stomach and duodenum and small intestine and so on. So, if we take a look at this figure, of course we can start from the beginning, from the events which occur in our mouth. And regarding digestion of proteins, actually nothing is going on. So only mechanical ground grinding of food and mixing with saliva and preparation for the gastrical phase of digestion. So digestion actually starts in the stomach and ends in small intestine. But what is important and interesting here is that enzymes are synthesized in stomach or in pancreas in the form of inactive precursors, which are also called as zymogens. Usually, those are much larger molecules comparing to their active forms, So activation is actually completed by the removal of the certain amino acid sequence in order to get an activated molecule, which is of the smaller molecular mass. This activation occurs in gastrointestinal tract lumen. So we have a bunch of enzymes: pepsinogen synthesized by stomach, trypsinogen, hemotrypsinogen, proelastase, procarboxypeptidases synthesized by pancreas, aminopeptidases synthesized by enterocytes, and so on. Notice that for inactive zymogens, we are usually using either suffixes OGEN or prefixes PRO to mark that it's an inactive precursor. But it is important to emphasize at this moment that no single enzyme can completely digest a protein molecule. More obvious, it's going to be a concert activity of several enzymes. So by coordinated action of all of these digestive enzymes, complex, huge protein molecules are going to be degraded into smaller peptides and amino acids, and then further on, smaller peptides are going to be degraded to amino acids by the action of enzymes synthesized by enterocytes. At this stage of digestion, at this moment of the story, we should maybe mention Kwashiorkor syndrome, which is observed in children in third world countries, which is developed as a consequence of deficiency of proteins in diet, especially deficiency of essential amino acids in a diet, with an adequate or inadequate calorie intake. In this case, muscle wasting occurs, and due to decrease of protein intake, also there is a decrease in the blood protein content, focusing on the decrease in blood plasma albumin. And if you remember, one of the functions of albumin is binding of water and maintaining the volume and oncotic pressure of blood. So if there is a deficiency of albumin in blood, water molecules are going to diffuse through membranes, and they're going to transfer to interstitial fluid. So there is going to be an increase in interstitial fluid observed. So it develops edema and so-called plump distension of abdomen and some kind of plump abdomen, which is very specific, having in mind that those children are very, very, very slim, with the lack of muscles on their extremities, but with those plump bellies. So anyway, let's start with the events in the stomach. So first, hydrochloric acid is synthesized by parietal gastric cells, and it's secreted by active transport, this time by hydrogen potassium ATPase pump. And the role of hydrochloric acid as a strong acid is to denature proteins, which means that upon the action of hydrochloric acid, proteins are gonna lose their upper types of protein organization, so the quaternary structure, tertiary structure, secondary structure, but no hydrolysis of peptide bonds themselves occur. So actually, the action of hydrochloric acid is to prepare the protein for the action of further digestive enzymes and to expose peptide bonds to those digestive enzymes. So the enzyme, the major enzyme which is active at the stomach level in gastric juice is pepsin, very famous one. It's an acid-resistant endopeptidase, which means that it cleaves peptide bonds inside the protein molecule. Pepsin is synthesized by chief cells and secreted in the form of inactive pepsinogen. The activation is completed by the autocatalytic function of pepsin itself. So it can cleave itself, so it can remove its own part of the molecule, and in the presence of hydrochloric acid, it can be converted to its active form. So the optimal pH for the activity of pepsin is pH of the gastric juice, 1.5 to 2.5. The catalytic function of pepsin is degradation or hydrolysis of proteins to smaller peptides, and rarely free amino acids are released at this point of digestion. Speaking about the specificity, it's rather low, and most of the authors claims that it's rather low. So pepsin actually randomly cleaves peptide bonds inside the protein chain. But some of the lecturers claim, and now I'm telling you because of the test, that pepsin is kind of specific, which is not exactly the truth, but there are some data suggesting that maybe pepsin has a slightly higher affinity to cleave peptide bonds in which a carboxyl group of aromatic amino acids and acidic amino acids were involved. But not that they are specific to them, but just slightly higher affinity to them. If pH of gastric juice rise from above 7 for many reasons, irreversible inactivation of pepsin occur. One more enzyme active here worth mentioning is chymosin in newborns, which is responsible for the digestion of milk protein, casein. Now we're moving on to duodenum. So the first event taking place there is neutralization of acidic pH of gastric juice due to the secretion of bicarbonate ions. But there is one more role of those bicarbonate ions, not only to neutralize acidic pH of gastric juice, but to set the optimum pH for the action of pancreatic enzymes. This stage of digestion is hormonally regulated by hormones cholecystokinin and secretin, but I'm not going to go again into details because we have explained the hormonal regulation of this phase of digestion in our previous videos. So when secreted in the form of inactive zymogens, pancreatic enzymes have to be activated in duodenal juice. Speaking of their activation, trypsinogen is going to be the first one to be activated and converted to active trypsin by the enzyme enteropeptidase from the brush border cells. This enteropeptidase actually removes one hexapeptide part in order to activate and expose the active site of trypsin. Then, activated trypsin activates all other enzymes. So hemotrypsinogen is going to be converted to hemotrypsin, proelastase to elastase, procarboxypeptidase to carboxypeptidase, and so on. It is worth mentioning, and at this very point, that pancreas also synthesizes a trypsin inhibitor, which is responsible to keep trypsinogen in its inactive form while in pancreas, in order to prevent the accidental activation of trypsin, which might take place in pancreas, which would actually cause the subsequent activation of other proteases and autodigestion of pancreas, which is one of the causes of pancreatitis. So, speaking about these enzymes, Pancreatic enzymes active in duodenum can be classified in two large groups. Endopeptidases, so cleaving peptide bonds inside the polypeptide chain, and exopeptidases, cleaving peptide bonds from C and N terminus of the polypeptide chain, thus removing amino acids from peptide chain's ends. Which enzymes are considered to be endopeptidases? Those are three pancreatic enzymes: trypsin, chymotrypsin, and elastase. So trypsin actually breaks down peptide bonds in which carboxyl group of arginine and lysine was involved. Chymotrypsin cleaves peptide bonds in which carboxyl groups of aromatic amino acid and leucine took part, and elastase cleaving peptide bonds in which carboxyl groups of alanine, glycine, and serine took part. So proteins which cannot be hydrolyzed by those endopeptidases are considered to be indigestible. And I'm going to give you an example like keratins. Of course, there are some inhibitors of those enzymes, and I gave you just one example without an intention to go into details. Diisopropyl fluorophosphate, which is an inhibitor of all those so-called serine proteases and so on. Speaking of exopeptidases, the one which is synthesized by pancreas and secreted to duodenal juice is carboxypeptidase, which actually removes C-terminal amino acids, and there are two types of carboxypeptidase. Carboxypeptidase A, which removes non-polar amino acids, alanine, valine, leucine, isoleucine, and carboxypeptidase B, which removes arginine and lysine. But there are some other enzymes active, which role is to complete the digestion of proteins which are active in both duodenum and small intestine, and all of those are exopeptidases, but they are synthesized by intestinal epithelial cells. Among those, we should mention aminopeptidase, which is responsible for removal of N-terminal amino acids within the brush border. And there are tripeptidases, dipeptidases, which cleave tripeptides, dipeptides into amino acids. So as a final result of the digestion, all dietary proteins are digested to amino acids. But autodigestion also occurs, and those proteolytic enzymes, they digest each other, so there is a digestion among themselves. And also there is a digestion of intestinal cells which are regularly slowed off into the lumen, also taking place. At this point of digestion, maybe we should mention one of these enzymes and illustrate that this one is found at some other points in our body but gastrointestinal tract, and that is elastase. Elastase is an enzyme which can also be found in neutrophils, which are responsible for destruction of invading bacteria and viruses. And often there is a certain quantity and amount of neutrophils in lungs, and sometimes neutrophils release certain small quantities of elastase. But in healthy individuals, this elastase, released elastase, is blocked by one protease inhibitor, which is called alpha-1 antitrypsin, which is synthesized and secreted by liver. And due to some genetic mutation, inactive alpha-1 antitrypsin can be produced, which actually yields to proteolytic destruction of lung cells and development of emphysema. Moving on to small intestine, we have completed digestion, so we have the mixture of amino acids which can be absorbed to enterocytes to intestinal brush epithelial cells. So absorption of amino acids is completed, as you can see from the figure, by active sodium-dependent transport, which is actually co-transport of amino acids with sodium ions. And this one is enabled due to the activity of sodium-potassium ATP pump, which actually provides negative gradient of sodium ions in intestinal epithelial cells because of the secretion of sodium. So the lack of sodium is actually replenished by the sodium from intestinal lumen, and amino acids are co-transported together with those sodium ions. This is very important because by the action of this active transport, it is possible to concentrate amino acids inside the cell from intestinal lumen. The mechanism which is involved in the further secretion of those amino acids to portal bloodstream is facilitated diffusion. So this mechanism is very similar to the one which we've illustrated as the absorption mechanism for glucose molecule, if you remember. But regarding amino acid absorption, there are at least 7 different transport systems which are specialized for different groups of amino acids. So certain transporters are specialized for neutral amino acids, some for basic amino acids, some for acidic amino acids, and so on, maybe neutral or aromatic. And of course, there is some certain overlapping in those amino acid groups. So for most of those amino acids, there is more than one transport system responsible for the absorption of that amino acid. Speaking about the disorders of the amino acid absorption, we can mention and illustrate the problem with one example, and that is Hartnup disease, which is actually inherited defect of neutral amino acid transporter, which means that this disease is actually developed as a result of the defect of essential amino acid transport, and it can be expressed in numerous ways, from asymptomatic to showing various clinical effects. Some of the patients develop so-called pellagra-like symptoms, which are very similar to pellagra, which is developed as a result of niacin deficiency or tryptophan deficiency, and manifested by symptoms like rash, ataxia, and psychiatric symptoms because if you know, tryptophan is necessary and it's a precursor for serotonin production. But what about the amino acid entrance into other tissue cells? It's completed by, again, active sodium-dependent transport, which is different, for example, from the entrance of glucose into tissue cells, which was accomplished by facilitated diffusion and GLUT transporters. So this time it's an active transport, and again, by this active transport, cells are enabled to concentrate amino acid inside and form so-called amino acid pools, from where, of course, amino acid can be used for various purposes. But apart from those dietary proteins digestion, tissue proteins are also constantly degraded in our body. So each tissue protein has its own half-life, which is a time at which 50% of the protein that was synthesized at a particular time will have been degraded. So a half-life can be rather short, from 5 to 20 minutes for some enzymes, up to several hours and days for some other proteins. But anyway, nevertheless, there is a dynamic equilibrium between the synthesis and degradation of tissue proteins, which is described by the term protein turnover. So the approximate daily turnover is 33 to 400 grams of protein, which is a lot, which means that approximately half of the kilogram of proteins are constantly being synthesized and degraded in our body during the day. So the total amount of proteins remains constant under normal physiological conditions. Out of those, 75% of amino acids are recycled, meaning they are used for the synthesis of new proteins, and 25% of amino acids are obtained from other sources, mainly food. An average daily intake of proteins ranges from 70 to 100 grams, and of course, there are some variation due to the different dietary habits. So there are some proteins, let me list and give you examples of some proteins with short half-life and very intensive protein turnover, like hemoglobin, like muscle proteins, like digestive enzymes undergoing autodigestion, and so on. It is interesting to explain muscle proteins phenomenon. Of course, you now know during fasting periods, we use glycogen reservoir for obtaining glucose and energy. Then we use lipids for getting fatty acids for beta oxidation. But when those sources are depleted, then our body starts degrading muscle proteins in order to obtain amino acids, which are going to be converted to precursor, which can be involved in gluconeogenesis. But now we are going to a very important point. After ingestion of proteins in diet, resynthesis of muscle proteins occur. So the muscle proteins are going to be replenished upon starting taking proteins in diet. But what is very important, that adults cannot increase the amount of muscle proteins by eating an excess amount of proteins. So it does not mean that you eat a lot of proteins, for example, a lot of meat, a lot of vegetables, plus protein supplements. You're not gonna increase the amount of muscle proteins just by eating protein-rich food. On contrary, all excess proteins which are not needed for the body at that specific moment are going to be converted to either glycogen, and we know that those are limited reservoirs of glycogen, or they're going to be converted to 3-acylglycerols and deposited in adipose tissue, which is an unlimited storage. So having this in mind, be very careful when making your diet plan in order to achieve your goals. This is something that we're gonna move on in our next video, but let me just explain you one phenomenon. Metabolism of amino acid yields the formation of nitrogen metabolites, various nitrogen metabolites, and we're gonna see those pathways in details in next video. And those nitrogen metabolites are excreted from the body by urine. So there is always certain loss of nitrogen from the organism due to this excretion. But nitrogen is also replenished by the absorption from gastrointestinal tract. So as a conclusion, we can say that the amount of nitrogen from diet is equal to the amount of nitrogen excreted in urine. And this is explained by the term nitrogen balance. So the total amount of nitrogen remains constant, like the total amount of proteins remains constant. During the phases and stages of growth and development, there can be positive shift. Positive shift can be observed, because apart from the turnover and recycling of proteins, there is a need to synthesize new proteins for growth and development. But negative shift is definitely considered pathological when we are losing more nitrogen than getting it from the food, and in those severe extreme cases it might be lethal as well. Degradation of those intracellular proteins is completed by the action of numerous enzymes, and I'm going to list here just some of those. Cathepsins are worth mentioning, A, B, and C, and those are endopeptidases degrading tissue proteins. And then we have different caspases, matrix metalloproteinases, proteasome, which actually acts through one small molecule called ubiquitin, one small peptide molecule, which actually targets the protein to be degraded by proteasome. Serine proteases, and so on and so on. So, my dear colleagues and students, I hope this topic was simple and easy and interesting if possible. So now we're gonna move on to some other topics regarding amino acid metabolism, ammonia metabolism, and so on. So until next time, stay healthy, stay safe, and bye-bye.